and thank you for the invitation to talk here. So today I want to cover some of our work on cytochrome C. Talk is titled Deciphering the Dynamics of Ferrous Cytochrome C Photodissociative State using Ultrafast X-ray Spectroscopy and Scattering Methods. So I'll start out by giving a bit of background on cytochrome C, starting with the biological functions the protein takes part in, as well as previous ultrafast measurements that have been performed on this system. And then after that, I'll go into the work that we've done, mostly focusing on ultrafast X-ray spectroscopy and solution scattering of ferrous cytochrome C. Talk a bit about the changes in spin state and local structure in the iron upon photodissociation. We'll also talk a bit about how heat dissipation occurs from the active site into the bulk. And we're going to use all of this to quantify what is known as the protein entatic state. And I'll define what that is shortly. So I'm sure most people here have heard of cytochrome C. It's a very important protein that plays roles in functions involved in life and cell death. So cytochrome C is most known as an electron transfer protein. And there it helps shuttle electrons between complexes 3 and 4 in the electron transport chain. When cytochrome C is performing this function, it has an active site structure that is the native structure to cytochrome C, which is what most people would probably be familiar with. It's 6-coordinate low spin, and this can shuttle easily between ferrous and ferric iron. This methionine bond is here, and the lack of structural change between the iron 2 and iron 3 states helps to cut down the reorganization energy for this protein, and this allows it to function very well as in shuttling electrons. However, more recently, it's been discovered that cytochrome C also plays a role in cell death or apoptosis. And basically the way this works is you have cytochrome C interact with something known as cardiolipin. Now, when cardiolipin interacts with cytochrome C can function as a peroxidase. And I'll avoid the chemistry involved in this. Long story short, this allows cytochrome C to escape from the membrane, and this triggers cell death. But the key point here is that the cytochrome C will function as a peroxidase, and this requires that one of the axial sites be open. Specifically, the methionine will be lost. This is the only way that the cytochrome C can function as a catalyst. So you have a protein now that's well optimized for 2 different functions. However, each of those functions requires a completely different coordination environment for the iron in the active site. So in a previous experiment that was done by Thomas Kroll, cytochrome C, both the ferrous and ferric forms, were studied using K-alpha resonant inelastic X-ray scattering. And the idea here was they would model the Rick spectra, which give you an L-edge-like spectra since you're looking at the K-alpha Ricks. Use this to extract the differential orbital covalency, which is the amount of metal D character in each of the iron D orbitals. And with this allows you to tune DFT calculations to get pretty reliable thermodynamics out of the system. What they found after they did this modeling by DFT is that the enthalpy of the iron sulfur bond in the ferrous form is extremely weak, only 2.6 kcals per mole. And this is actually less, this is small enough such that you would expect the methionine to be deligated at room temperature. I believe T delta S at room temperature here will be about 4.5 to 5 kcals per mole. And there's an obvious issue with these numbers. We know that cytochrome C functions in electron transport where the methionine is bound, yet thermodynamically we see that the methionine probably shouldn't be bound. And that's because we're not taking into account in these calculations the role that the protein plays in keeping the methionine bound, specifically the amount of energy that the protein dedicates to the iron-methionine bond. And this is what we call the protein entatic state. Basically, the protein bulk is compensating for the inherently weak bond that the methionine forms to the iron 2 center. So how can we get at figuring out how all this works and perhaps even quantify this protein contribution? Well, it's cytochrome C, both the ferrous and ferric forms, have been studied using ultrafast transient optical transient absorption measurements in the past. And one thing that's found out, and this tends to be consistent across, I believe, all ferrous heme proteins, is that when you photoexcite this system in the heme pi to pi star bands, you either excite at the main Sorey band or the alpha beta bands down here, it doesn't matter. Either way, when you photoexcite, transient absorption, you observe a strong ground state bleach along with a strong cytostate absorption. This is the case when you pump at the alpha beta bands or at 400 nanometers of the Sorey. Either way, that strong cytostate absorption suggests that you've changed the structure of the system. Specifically, you formed a 5-coordinate high spin heme system. And alternatively, interestingly, in the case of ferric cytochrome C, you don't observe a strong cytostate absorption out here. Instead, you see a very strong ground state bleach, which suggests that when you photoexcite this thing, you haven't lost a ligand, but instead you've probably just dumped a lot of external heat into there that relaxes in some time scale. Additionally, these systems were investigated using time-resolved resonance Raman. And we're not going to go into breaking down all these vibrational bands, but the key one is right down here. This is observed in deoxymyoglobin and hemoglobin. And this is an iron histidine stretch, the other axial ligand, but it only shows up in a 5-coordinate heme. It's observed in ferrous cytochrome C, but not the ferric. And this suggests, again, that photoexcitation in the heme pi to pi star bands of ferrous cytochrome C causes loss of the axial methionine ligand. Now, the time scale for this axial loss this occurs on the sub-picosecond time scale, and the lifetime of cytochrome C, when you photoexcite it, is in the single to maybe 10 picosecond range, which is much too short to capture using synchrotron X-ray radiation, where you have intrinsic time width of about 100 picoseconds based on the X-ray bunch width. So instead, we decided to look at these systems at the LCLS, where you can achieve up to sub-100 femtosecond time resolution if you desire. So we get time resolution sufficient to actually probe these systems. And initially, we decided to look at these systems using both iron K-edge X-ray absorption to quantify some of the electronic changes, but primarily to look for structural changes in the shape resonances post-edge. And we also used Kbeta X-ray emission. Now, Kbeta X-ray emission is a very good sensor of the metal spin state. You may recall a paper from 7 years ago now that tracked intermediate spin states in ferrous iron tris-bipyridine, basically trying to use the same sort of methodology now. And we'll go into the specifics of the Kbeta emission shortly. But we can start with the iron K-edge XAS. So as we said, we expect upon photodissociation that we will lose methionine ligand. This will also cause an increase in the axial histidine distance, as well as an increase in the iron to porphyrin nitrogen distances. And the increase in the equatorial bonds is expected to be a product both of heme core expansion, as well as propagation of the metal center out of the plane of the ring. What we have up here are the ground state iron K-edge, measured at about 120 to 140 EV past the rising edge. Ground state shown in black, and in this case, we probed at 600 femtoseconds, which is shown in green. 600 femtoseconds is chosen simply because that's where the maximum difference signal in the transient absorption is observed. And there's a couple things that we noticed right away. First, we have a shift in the white line energy to lower energy, which is something that can be observed in these cases with a change of spin. In some cases, a shift in the edge like that could indicate an oxidation state change. However, that can also be indicative of spin state changes. Additionally, we also have changes in what we call the shape resonances out here. And as we know from metal K-edge spectroscopy, a lot of these low energy features at low energies past the edge originate from multiple scattering contributions, basically from the iron. So what we wanted to do is try to extract the structure from these. Here we go. All right. And so we used a program called MXAM, which is designed for simulating multiple scattering spectra. And this is shown down below. So on the bottom left here, we have experimental data for the ground state, along with a fit shown in blue. And this is fit to a basic DFT model, just an optimized singlet ground structure. So the way these simulations work, I'm not going to go into a ton of detail here. But basically, you have a set of coordinates, you have an interstitial potential, and you also have muffin tin radii, along with another number of other potential variables. And you can optimize all these and reproduce the shape resonances based on your structure. So there's a peak on the rising edge that we miss because this is some kind of p symmetry electronic transition, but we generally reproduce the shape of the zanes quite well, including the little shoulder here and the general intensity of the two peaks that are out here. Alternatively, we can also generate a model for the high spin ferrous cytochrome C, which is shown on the bottom right here. And again, we reproduce the general shape of here pretty well. Now, this DFT model has elongation of the iron histidine distance. It goes from around 2 angstroms in the ground state to 2.15 in the excited state. We have 2 angstrom elongation to 2.09 angstrom roughly in the iron to porphyrin nitrogen distances, as well as an extremely large change in the iron-methionine distance, which is basically just telling us that the methionine is gone and the methionine isn't contributing significantly to the model here. So I exclude for brevity here other models involving different axial distances, since this is the best fit here. But the long story short is that we observe sort of what we expected, which is that when we photoexcite this species, we adopt a high spin iron center, and we have an appropriate structure for that, where we lose the methionine axial ligand, and we have expansion of the iron equatorial bonds along with expansion of the iron to histidine bond. Okay, any questions so far?
I'll interject just a quick one. I realize you haven't gotten into the details of LCLS and whatnot, but what is your photoexcitation spectrum, for example, in the study you just showed?
The photoexcitation spectrum optically you're talking about?
Yes, optically.
So we don't have it shown here, but basically we reproduce the general changes that are observed in the literature, right? So we excited at 520 nanometers, which is one of the vibrational bands this is the alpha band basically. Mm-hmm. And when you photoexcite there, you get a bleach, at this case in the peak of the Sorey, but you also get an excited state absorption farther into the red.
I guess what I'm asking is in the LCLS experiment, are you fully bleaching all of the cytochrome C in your liquid jet?
No. That's a good question. And I wish I would have included a slide about this. So one thing that we want to avoid is multiphoton absorption of the optical beam, right? So you can increase the power indefinitely. However, at some point you're going to lose. It's a basic Beer's law argument, right? At some point you lose linearity of the absorption of your photons versus the excited state fraction that you generate. At that point, you have multiphoton absorption, which could have strange effects on the sample as well as the bulk solvent potentially. So what you do is you do what we call a power titration. In this case, basically we looked at the intensity of the K alpha emission as a function of the laser fluence. And then you cut off right before the end of the sort of linear regime there. And this gives us around a 70% excitation fraction. So you have around 30% of your sample that remains in the ground state. You can achieve these sort of high excitation fractions because the absorption coefficients of the porphyrin optically are very strong. For ferrous cytochrome C at 520, this is the weakest of the main bands, and this has about a 10,000 per molar per centimeter excitation fraction, which is quite large. So yeah, you can typically, with very high powers, get well into the tens, into the tens of percentage of excited state fraction, which we do here. And for reasons that we'll discuss later, in particular, understanding sort of the thermal dissipation from the active site, it's really important that we know that everything we're observing corresponds to one photon absorption, or 1 photon per absorption event. To say, and generally, ensure that with the power titration. Mm-hmm.
All right, very good. Yang Ha, you have a question?
Yeah. Hi, Mike. Some detail. Yeah, just the slide. You talked about the DFT on the 2 states, especially for the high spin state. Did you optimize it to a ground state or an excited state?
To a ground state.
So you basically force the spin and you just let it optimize.
Exactly. So that works all right here. I'll get to this a bit later, but it had been speculated up to this point that that upon photoexcitation on this, what you actually see decay on the picosecond time scale is actually from a vibrationally hot 5-coordinate ground state. And so doing these simulations on basically the quintet ground state in this case is sufficient to model these complexes.
And what's the energy difference between these two states?
Let's see, I believe that from DFT, these are separated by about 6 kcal per mole.
Oh, okay. Thanks.
Mhm.
All right, seeing no other questions, you should continue.
Okay, great. Okay, so next, let's talk about the Kbeta X-ray emission measurements. So of course, at the iron K edge, you put in a monochromator, you excite on resonance. With the Kbeta emission, we don't do that. Instead, we set the emission energy or set the incident energy at about 8 keV. And we measure emission of the as the 3p electron falls down to fill the 1s core hole. This ends up being around 8 to 10 times weaker than the K alpha emission, which would collect the 2p to 1s emission from that transition. However, in the K beta, you have strong coupling between the 3p hole and the 3D manifold. And what ends up happening as a result is you end up getting very sensitive spin state information. The other advantage to looking at the Kbeta emission versus the XAS is that with dispersive optics, which I've shown down on the right, you can collect your full Kbeta emission spectrum as a single shot. So if you want to look at the dynamics as a function of time, it's a lot easier to map out the emission over multiple picoseconds worth of time than it would be for the XAS, where you'd be stuck at single absorption energy. So for that reason, we first look at the recombination of the methionine ligand, and we'll do that using the Kbeta X-ray emission. So experimental emission spectra are shown at the top. Ground state again is in black, and for consistency with the XAS, I show 600 femtosecond time delay here. So in the singlet state Kbeta emission spectrum, you see the single mainline peak. However, when we photoexcite this, we see both a shift in that mainline peak to higher energy, but you also see this additional band that shows up around 10 to 15 EV below that. Now, as I said before, these emission spectra are quite sensitive to the metal spin state. As you go from the singlet state all the way up to the sextet state, you start to see this steady shift in the white line peak, and then you also see this satellite peak grow in. And the satellite peak is a function of the exchange interaction between the p hole and now the mostly spin aligned d electrons. And knowing what the structure of these spin states are, we can easily track the recombination of the methionine to the iron in cytochrome C by looking at the decay of the quintet state versus the recovery of the singlet state. And when you do this, you see that we have roughly a 5 to 6 or so picosecond time constant for the methionine recombination to the iron. Now, as I mentioned before, Kbeta X-ray emission had been used in previous measurements to identify a spin intermediate in ferrous iron tris by purity. Okay, this is a pretty well-known spectroscopy paper from the LCLS, so a lot of you are likely familiar with this. And this was tracked using Kbeta X-ray emission. Essentially, you see changes right around the mainline peak that grow in on a different time scale than the satellite feature grows in here. So while we've mapped out the long time dynamics of our system, we haven't really commented on the potential intermediate spin state, which could live for maybe 100 or so femtoseconds and then decay out. And so that's what we sought out to do. So the analysis of this data, as well as scattering data later, was done by Marco Reinhard. And what we have down here is sort of early time points in the evolution of the emission of the Kbeta emission spectrum. At negative times before the laser pulse arrives, we have a flat line, which is always good as a sanity check, right? And then these dotted lines here show these two different kinetic traces here. And one thing that you'll observe, and you can observe it in the 2D data too, is that you have this portion of your Kbeta emission spectrum that's clearly growing in on a different time scale than the different signal in the mainline peak. This blue feature being this negative feature here, the shift in the mainline to higher energy. So these different kinetic traces here all suggest that we have an intermediate spin state in this system. And so with modeling, you can extract this. This is a model of the experimental data. You see it fits quite well, and it's using a population analysis shown here. So in our experiment, we have 120 femtosecond time resolution, full with half max. This is largely dictated by the 100 micron liquid jet that we use, since you have a laser and X-ray velocity mismatch going through the jet. And so initially we pump into a porphyrin pi to pi star state. Now, these X-ray spectroscopy measurements are not sensitive directly to the porphyrin pi to pi star transition. However, these have been nailed down pretty well optically. So from fluorescence upconversion measurements, this is known to have about 145 femtosecond lifetime. Now, we have a growth of what is identified as a triplet metal centered state, when this is a lifetime of around 90 femtoseconds. And despite our time resolution, you're still able to distinctly resolve this triplet state. The triplet state decays very quickly into the quintet state. Now, naturally, with the longer pi to pi star decay lifetime versus the relatively quick triplet decay time, you don't build up a large population of the state, but it is observable. And then again, we populate a quintet state on around a 6 picosecond time scale. And this decays directly back to the ground state. There's no observation of any other intermediate going from the quintet state back to the ground. Okay, so the Kbeta X-ray emission gives you pretty good spectroscopic evidence of the presence of an intermediate triplet state. However, it's not very good at identifying what the nature of that triplet state may be. So, in ground state Ferrous Cytochrome C, we'd have fully occupied dxy orbitals as well as d pi orbitals, and we know that at the higher spin state in the triplet state, one of these d pi orbital or electrons is going to be lost, and it's going to populate either the dz squared or the dx squared minus y squared. Now, logically, we may expect this to be the dz squared, being that this will be the lower energy orbital. However, we don't actually know that for sure, and the X-ray emission can't really tell us that. However, there are a few things that we do know. The dz squared orbital, of course, is going to be destabilizing with respect to the axial bonds. So if you populate the dz squared, you would expect elongation of the methionine and histidine ligands. Alternatively, if you occupied the dx squared minus y squared, you would expect this to be destabilizing with respect to these equatorial bonds, which would cause elongation of the iron to porphyrin nitrogen bonds. And so for these early time points, we decided to determine the structure here using X-ray solution scattering. Now, X-ray solution scattering, like the Kbeta emission, doesn't require you to be on resonance with the iron K edge. And there's a couple advantages to this. The first is that we now no longer require a monochromator, which is great because a monochromator causes us to lose a lot of flux. But again, since this is a dispersive technique, like with the X-ray emission, you can collect full spectra a single shot at a given time delay. If you want to scan through time, it sort of decomposes the experiment into a one-dimensional type of scan. So to investigate these early time structures on the first couple hundred femtoseconds following photoexcitation, we decided to use X-ray solution scattering. Okay, so up here is a 2D difference plane for the X-ray scattering. Down the bottom are individual time cuts. And you'll see a few things in here. First, we see a number of different signals that show up at very early time delays, this first line cut being 100 femtoseconds. And we see those starting to decay on the single picosecond time scale. But we're also seeing a new feature that grows in here between 2 and 2.5 angstroms. So we can basically qualitatively separate the scattering, the scattering signal here into two components. The lower Q signal, say from maybe here down, is going to correspond to structural changes in the protein active site. And at really low Q, you would also likely pick up changes in the protein bulk, although we're not doing that here. At larger Q values out here, we're picking up the solvent response. So the scattering signal will pick up changes that occur in the solvent, both as a result of changes in its temperature, as well as changes in the position of the molecules, or the density, if you will. So for right now, we'll focus on the low Q portion of this data. and the modeling there. These black lines shown here show fits. And basically, the time-dependent scattering at Q below, I believe it was 1.2 inverse angstroms, can be modeled very effectively simply by moving the axial ligands out. So you see, you start at just below 2.4 angstroms for the methionine, and that expands out past 2.6 angstroms on the first couple hundred femtoseconds. And alternatively, you also get the expansion in the iron histidine bond length at very early times. So the data can be effectively modeled with only axial distortions without having to include any equatorial expansion. And what this suggests is sort of what we expected initially, which is that we, in the triplet state, we have an occupied DZ squared orbital. This is likely the dissociative state for this system because it's the one that's going to destabilize the methionine bond. Methionine flies out, and we also get elongation in the iron histidine bond when we occupy this triplet state as well. Okay, so I guess we can stop for questions now.
We'll give people just a second. I'll ask a general question. So the combination of the spectroscopy and the diffraction, is that becoming more common in these kinds of photoexcited problems?
I think so, and partly because the X-ray emission is really well catered from a spectroscopy perspective, I think, to these LCLS, these XFEL measurements in general. And again, I think the reason is, well, there's a few things. One, you don't have to change energy, right? So you can exclude a monochromator, you get more flux. Changing the incident energy is, at least, especially back when these measurements were done, was not trivial. You get a broadband output from the self-amplified stimulated emission, which is what we use to cover the zanes. But there's a big loss in signal because you are scanning over energy through a monochromator, right? Whereas the X-ray emission, you sit at an incident energy, you never really have to change that, and since it's dispersive, you can scan that through time. And the geometry of the experiment means that you can put a detector in for the forward scattering without any problem. So there's really no reason not to do these separately. The scattering basically ends up being a pure structural measurement of your system, while the emission, in this case the Kbeta, is basically a pure electronic information measurement. And in principle, if you wanted, you could also do the valence decor emission on the high-end shoulder of the Kbeta emission. We didn't do that here, but that's something that's accessible. So that seems to be an ideal way to measure these kinds of systems, and it's been used for a ton of non-protein transition metal type complexes as well.
Okay. Galen, I see you have a question in the chat. Would you like to ask?
Yeah, can you hear me?
Yes.
I said, can you clarify what you mean by exclude a monochromator to get more flux? I'm pretty sure you just mean you're not monochromating the incident beam, and you're just, there's some jitter and there's some width in the beam. And can you tell me what those values are?
Okay. So I think in the iron K-edge XAS measurement that we presented here, we had a standard silicon 111 monochromator. I do not recall the bandwidth of the beam without the monochromator. It's certainly EV, which is problematic for from an X-ray absorption measurement. At 8 keV or even at 7.5 keV, we would be more than sufficiently off of the iron K-edge to really have any issues with the jitter that we need to correct for. But I don't really remember the specific specs beyond that. But yeah, what I basically mean is that with XAS, you put in a monochromator and this attenuates your beam substantially, right? Whereas your full beam, you can take advantage of the increased flux to collect these other pieces of data much more quickly.
Yeah, that makes sense. Thanks.
Mm-hmm.
Okay, seeing no more questions, you should continue.
Okay. So one question that came up is how you hop over from the triplet state onto the quintet state. So shown on the left here are basic DFT calculations. So what this is here is we start from our singlet ground state and we simply pull our axial ligands out. When we pull our axial ligands out, the energy of the triplet and quintet species in those structures would both drop, right? However, the quintet species has an electron in the d squared minus y squared. It wants equatorial expansion. And the result of this is the triplet and quintet potential surfaces will never cross if you're only including axial ligand expansion. And this would seem to be a bit of a problem, right? You see that the primary changes that we're observing are really are primarily among the z-axis. So how would it be possible in principle for the triplet to cross over into the quintet? And one thing you have to keep in mind here is that in this triplet state, we have a hole in this d pi orbital, right? So we have sort of asymmetrically occupied xz and yz orbitals. Now, this d pi hole will have a specific alignment. In an optimized triplet structure, these orbitals will be aligned very well in between the x and y axes. However, what we find out in DFT is that initially, when we calculate this triplet state, this d pi hole is actually aligned along either the x or y coordinate. So basically along one of these iron-nitrogen porphyrin bonds. And what result this will have is this will cause sort of an asymmetric expansion of the iron porphyrin bond distance. It'll be small, but you'll have some expansion either along the x or y axis. And this actually lowers the energy of the quintet relative to the triplet enough such that they can cross. And so from all of this, you can put together a general mechanism for how this photodissociation process and the subsequent recombination occurs. So basically what we have is we have the initial excitation from the pi to pi stars. We're in some singlet porphyrin state, right? And eventually we manage to cross over into the triplet state. This is what we observed in the Kbeta X-ray emission. And we have expansion of the axial distances, but also this initial alignment of the d pi hole along the we'll call it the x coordinate here. So you'll get very minor expansion in these equatorial bonds. So this allows the triplet state to quickly cross into the quintet. Now, once you're in the quintet state, you then have the expansion of the equatorial bonds, which is what we observed in the iron KHXAS models at 600 femtosecond time delay. And then again, from the Kbeta emission, we know that on the time scale of 6 to 7 picoseconds, we eventually cross back to the quintet state, back down into the ground state manifold. The methionine will rebind, and we will relax back down to just the standard ground state. So this is pretty standard in terms of the mechanism. The thing that we don't know from this is the precise mechanism to hop from the pi to pi star state onto the triplet manifold. And that will be the subject of future studies. We're not going to talk about that now because we don't have experimental information to really validate a specific model. But the last thing I wanted to talk about, I mentioned this earlier, was the heat dissipation from the protein into the solvent. And we saw in the scattering data that on the certainly the 10 picosecond time scale or so, we start to see a heating signal in the solvent. So the solvent scattering signal can be roughly measured by broken into two components, one that we call a temperature component and one a density component, right? The solvent will heat up, and then at later times, the solvent will start to rearrange itself. Now, with cytochrome C, we're looking at sub-picosecond out to single to tens of picosecond timescales. Right? On that timescale, the density changes in the solvent can generally be ignored. And so this breaks down into a simple that the argument that the scattering difference signal is going to be based on the change in temperature, which is to say, using the magnitude of the difference signal that we measure in the solvent, we can quantify the heat dissipation from the active site into the bulk solvent. Okay, so return to our scattering data we looked at before. So shown on the bottom left is singular value decomposition of the low Q data at longer time delays, basically the single to 15 or so picoseconds. And we can model this basically with one scattering component. Basically what this allows us to do is measure the decay in changes in the active site scattering signal as a function of time. And we get that around a 5 picosecond time constant for decay of the active site scattering signal, which is pretty consistent with what we saw in the Kbeta X-ray emission. Now, we can do the same sort of thing with the high Q signal, which again measures the solvent. And on the right here, we see that the solvent decay rises in with around a 7 picosecond time constant. This is pretty consistent with the scattering of the active site, as well as what we've picked up spectroscopically. However, a lot of work has been done on the magnitude of the different scattering signals and how they'll relate to temperature. So you can take these scattering magnitudes and we can actually relate them into the energy that's dissipated out of the active site, which is what's plotted over here at delta E. Additionally, we know what energy was lost from the active site because we know the amount of energy we put in. It's based on the pump wavelength, which was 520 nanometers, so 2.4 eV pump beam. And we know the excitation fraction, which was 0.74 in this case. So we can also quantify the amount of energy lost from the active site. And we see that this is all very internally consistent. So we have this ligand recombination of the methionine to the iron, and it coincides with the loss of the heat in the active site that goes into the protein. As I said earlier, this has been modeled as the 5-coordinate species has been modeled on this time scale as a hot ground state, essentially. And this makes sense, right? We form a 5-coordinate state with a bunch of extra vibrational energy in it, and that energy dissipates, it goes out into the protein ball. So one of the things that this suggests is that the methionine recombination may actually be dictated by a thermal equilibrium, right? We know that the bond strength is pretty weak, so at higher temperatures, we'll shift the equilibrium towards methionine off. As the temperature dissipates from the active site, it becomes more easy to rebind the methionine ligand. And so to sort of figure all this out and to figure out what the actual bond strength or the bond enthalpy of the methionine is in the protein, all that we need to do is relate a equilibrium constant to an active site temperature. Now, the scattering gives us an idea of how much heat is lost from sort of the active site as a whole, but it doesn't really tell us much about in terms of quantifying an effective temperature. So we actually went to the previously published time-resolved resonance Raman data for this. And one thing that was shown back in the 80s is that you're going to have a frequency dependence in a resonance Raman measurement of these heme bands on temperature. And the reason they end up depending on temperature is due to anharmonic coupling with a lower frequency vibrational mode that at higher temperature starts to be thermally populated. So the specific model here is a bit beyond the scope of this discussion. But the long story short is we have the shift in vibrational frequency of Ferrous Cytochrome C when it's photoexcited. We know how much the frequency shifts on these single picosecond time scales. As a bit of an aside, with something like transient resonance Raman, you're generally limited to around a couple picosecond time resolution if you have good frequency resolution. It's a Heisenberg uncertainty principle argument. If you need to get around that, you would do something like a femtosecond stimulated Raman. So in this previous study, it was assumed that what you're looking at in the recovery of the ferrous and ferric cytochrome C was actually a thermal decay, like we see here. But we have these frequencies associated with the local heating in the active site. We also have a way to convert them into temperatures. And if you recall from before, the Kbeta emission gives us the amount of quintet and singlet species that are remaining in the active site as a function of time. So we effectively have equilibrium constants too. And what this allows you to do is we can put together a standard Van Hoff plot, the natural log of the equilibrium constant versus temperature, right? At each of our time points from 2 picoseconds out until we have recovery. And we can do this, and this allows us to extract the enthalpy and the entropy of this, in this case, the iron-methionine bond in the protein, okay? Now, recall from DFT that we picked up an iron-methionine bond of about 2.6 kcal per mole in the ferrous cytochrome C. This was, of course, ignoring any contributions from the protein. But now, experimentally in the protein, we picked up an enthalpy of 6.5 kcals per mole, about 4 kcal per mole higher than what we picked up by the DFT. What this suggests is that the protein is actually contributing about 4 kcal per mole of stabilization to the iron-methionine bond. Now, if you work out the numbers here, you'll see that at room temperature, a delta H of 6.5 kcal per mole and an entropy of about 16 entropy units will give you 96 or so percent methionine bound in ground state. So within error, it seems like we've done a pretty good job here of identifying the specific sort of stabilization that's going on. Now this raised a question in my mind, which is, okay, great. Where exactly does this stabilization come from? And one interesting thing you notice if you look through, there's numerous different variations of cytochrome C. However, not all of the amino acid residues are conserved across all of these species. Things like the methionine and the histidine that bind to the iron are. But what else is conserved is things like this tyrosine, threonine, and asparagine that are all in this hydrogen bonding network with the methionine. So my speculation is that this additional 4 kcal per mole is actually coming from this hydrogen bonding network inside the protein. Now, one of the interesting things with cardiolipin is cardiolipin, shown up here, has a number of different docking sites in which it can insert itself into the protein. Remember, cytochrome C acts as a peroxidase of cardiolipin, which means that the cardiolipin has to access the ion center at some level. And one of the docking sites is actually the C site, which involves the threonine, asparagine that I just mentioned in that hydrogen bonding network. And so what I propose here is that the additional protein stabilization is from this hydrogen bonding network, and that once you come into contact with cardiolipin, it inserts into the protein and disrupts this hydrogen bonding network. This causes you to lose most or all of that 4 kcal per mole additional stabilization of the protein, and just based on simple thermodynamics, this would cause it to be lost just due to an equilibrium argument. So this 4 kcal per mole has been proposed in a number of other systems. I believe even there was a form of zeolite system that had by DFT a similar sort of stabilization from its sort of macrostructure there. But this here is the first experimental verification of a protein in entatic state. And this really helps us to understand how the protein bulk optimizes the active site to take part in 2 different processes that involve 2 completely different coordination environments. Okay, so just to briefly wrap this up, using sub-picosecond radiation from the LCLS, we looked at the ultrafast properties of ferrous cytochrome C, photodissociative state. We did this using ultrafast X-ray absorption and emission spectroscopies along with X-ray solution scattering. And we learned a few things. We learned the mechanism for photodissociation, namely that you proceed through a triplet state, that we know how you cross over the quintet state and eventually back to the ground state. We're able to quantify the structure. The quintet state structure is nailed down using the iron K-edge X-ray absorption, whereas at early times we see the axial elongation in the triplet species shown by the scattering. The scattering additionally allows us to track the thermal dissipation, which helps to further demonstrate that the recombination of the methionine is likely thermally driven. And using this information and being able to determine the temperature in the active site and using our equilibrium constant that we derive from the Kbeta emission, we can quantify this protein additional stabilization as 4 kcal per mole. As I said, this is the first direct experimental measurement of a protein in entatic state, as far as I know. And likely the way this works is cardiolipin interrupts a hydrogen bonding network. I'll be curious to see in the future as more studies focus on investigating the cardiolipin interaction with cytochrome C to see if this ends up being sort of verified. But again, one thing that we learn about the protein is that the protein manages to sort of put the bond strength energy into sort of a sweet spot, right? It's strong enough that it's stable under standard conditions for electron transfer, but it's not too strong such that interaction with cardiolipin can pull it back apart. It really just drives home how well tuned these proteins are and just natural systems in general to be able to perform multiple different roles that require completely different structures. So I want to acknowledge Professor Solomon, who was my advisor as a postdoc when I did the initial part of this work. Kelly Gaffney and his postdoc at the time, Marco Reinhardt, he published, he was the first author on the work, including the scattering and the early time triplet species, and did a lot of the analysis and a lot of the heavy lifting with the data processing. So he's quite good. Uwe Bergman and Keith Hodge and Britt Hedman also pitched in a lot in terms of ideas and how to proceed with experiments. And Roberto Alonso Moura was the primary beamline scientist at XPP, where we did this work. And I guess that's it. So if anybody has any questions, I'll take them now.
Yeah, people will be typing questions in. I'll start. If you go back to your conclusion slide for a second.
Yeah.
I asked earlier about whether the simultaneous spectroscopy and diffraction was becoming more common. Mm-hmm. Let me change that question around a little bit. If you didn't have the scattering, how far would you have gotten down this list of outcomes? I think you wouldn't have gotten the 4 kcal per mole. Is that correct?
Well, we could have still gotten the 4 kcal per mole, just based off literature that suggested that that we're looking at sort of heat dissipation from the hot ground state. However, that would have been a lot more speculative.
Okay.
Additionally, while we could have logically concluded that the triplet state was the dz squared occupied, just based on the energetics of and the splitting of the d orbitals, we wouldn't have had any direct evidence of that. Again, logically that makes sense, but it allows us to confirm that. But I think the scattering and the solvent response ends up being very important, because you have things like the time resolved resonance Raman that show that this was largely due to heat dissipation, right? But that's only directly probing the active site. So part of the utility of the scattering here is showing in a simultaneous measurement, right, that you're seeing the solvent response and sort of the protein response, and they sort of anti-correlated in a sense, right? So while you still could have come to the same conclusion, you could have extracted the same entatic state number, because you would have still had the same equilibrium constant argument from the Kbeta use, still would have had the same resonance Raman data. But I think that the scattering really nails home just how the heat dissipation works, and it's the only direct measurement of the temperature buildup in the solvent upon cytochrome C excitation that I know of. There's probably other ways to do it, but I haven't seen it if it's there.
Okay, thank you. Yang Ha, you have a question?
Yeah, hi, Mike. I have a general question. So yeah, about, can you make a comment on the role of the heme ring? Because all your system have the porphyrin ring. Like, can you make a comment on the role, like the ring constraint? And also, can the same technique be extended to other, say, non-heme ions or even solid state catalysts?
Okay, so that's a couple of questions. So there are constraints on the heme. For one, you're going to have reorganization of the heme in the high spin states, right? Just based on the occupation of the extra minus y squared. And you also have the side chains, like the two cystinate side chains that bind in the protein bulk, that probably have some sort of limitation on this as well.
Yeah, especially when you mention those like elongation in the z-axis, but pretty conserved in the xy plane. I think that's the
right. Now, it is worth noting that, of course, in the MET rebinding, we are looking at the conversion of the quintet to singlet species, which means that you should have that the heme core expansion should retract, I suppose, for lack of a better way to say it. It's not clear to me how that are you asking how that energetically affects like the entatic state?
Yeah.
Okay, because that I don't have as good of a feel for. Like I said, we pull out about a 4 kcal per mole stabilization from the protein, and that can mostly be accounted for, I think, based on the hydrogen bonding strength, which I've seen from OH groups to different neutral sulfur type groups is in the 3 to 4 kcal per mole range. In that respect, I suspect that what's doing the porphyrin ring doesn't affect it a lot, but I don't know how to comment on it much more than that. To answer your other question about whether or not this can be applied to other systems, I don't see why this sort of analysis could not be applied to other systems. The tricky thing, though, again, assuming you have a system where you have sort of this like a rebinding process that's dictated by a thermal equilibrium, like we have here, getting the amount of ligand on, ligand off, there's a number of ways to do that, right? While X-ray emission in this case is a very good way to do it because you're not sensitive to a bunch of other factors beyond the spin state. you could get it with a lot of error potentially from an optical measurement, I suppose. The tricky thing then becomes how you extract the local temperature. The heme was beneficial in that, the heme has a bunch of very prominent, very strong resonance Raman bands. And that porphyrins had sort of the specific model derived for how the frequency shifts as a function of temperature. And I think if you wanted to apply this to non-heme systems, I think the big key would be how you quantify the active site temperature there. Okay. Beyond that, the general methodology used in the X-ray spectroscopy and scattering should still apply. However, I don't know if it's straightforward to sort of translate the active site temperature as extracted from the scattering into a direct temperature in the active site. It's not something that I've looked into. In this case, it wasn't necessary. So it's hard for me to give you a better answer than that. But the hard part, I think, about this actually becomes how you define the active site temperature at these early times.
Very good. A last question, whether it's XFEL or LCLS2, how do you think that the higher rep rate is going to be influencing this class of study?
That's a great question. Well, I mean, it should certainly make experiments faster, right? We have a much faster scan rate, which is ideal. That may be better for picking up on a number of weaker features. One of the things I mentioned was something like the valence to core emission. And you can get a lot of very fine information about, the metal center and the bonding from that. However, it's extremely weak. So having much faster sampling could be better in that regard.
What about RIX or HERFT? Would they play could they play a significant role, or do you already have what you need just by characterizing spin?
The HERFT could certainly be valuable. And it would help you to clean up the pre-edge features for sure. And you may be able to get a lot of this information regarding the specific electronic configuration from the pre-edge peaks there, right? That's one of the big utilities in HERFT. I suppose if you have a cytochrome C itself is pretty easy as a protein. You can get a lot of it, and it's super pure. However, if you have a number of different proteins, you tend to get these background signals sometimes in the iron K edge signals. In steady state measurements, this has messed up a lot of different structural assignments. And one of the ways they clear that up is actually using HERFT. So it could help you in more complicated proteins. Certainly ones that have multiple metal centers, it might give you some ability to be more selective. And certainly something like HERFT or RIX, where you're pretty photon starved to begin with, could benefit in that regard. They said the cytochrome C and myoglobin or hemoglobin are pretty much the workhorse proteins for these sorts of things, right? Because you have so much of it, they're simple and they're very robust. You can get away with the slower rep rate and the slower sampling. However, you probably do benefit from more difficult experiments or sort of more elaborate proteins down the line. The issue that will come up, right, assuming that you have the same sort of fluence per shot, is that you'll probably destroy your sample a lot faster too. So this may now require a much larger sample environment. The nice thing with the low rep rate in our case is that even if you do destroy sample, it takes you quite a long time to actually reprobe the destroyed sample. So in your data, you never really see a buildup of decomposition product. But with a high rep rate, that will start to become a problem most likely.